Good evening, one and all, and thank you very much, Arun sir, for giving me this opportunity. And from the company, on behalf of ADrPLEXUS, on behalf of Pro MBBS, I welcome doctors, you one and all, for this discussion. Today's topic will be on nerve conduction. Before we get into the topic as such, let me be quick in emphasizing the importance of the reason why you got to start your preparation early and where we exactly, giving you the cuttage in shaping your career and your professional growth, people. So I hope you guys have had a idea of the questions that has been asked in NEET-PG entrance exam that took place on Sunday. If you'd have seen the questions, it's more of a very commonly asked questions, very easy topics, which has been conventionally getting repeated in entrance exams. But if you consider the mindset of students, once they come out of that exam hall, many of them were experiencing just one process, or many of them had got just one kind of review that they could able to narrow down two options, and they were not able to narrow down at the third and fourth options. And that is exactly where things changes. Your rank, your seat, your future, everything is dependent on those two options, people. So when it comes to the competition, when it comes to the rapid progress in the number of medical seats, it's pivotal and extremely important that you got to be very sharp in your subject, and you got to start your preparation quite early. And it cannot be very simple, it cannot be very primitive, all right? Your content, your idea, your subject knowledge can't be so superficial. It cannot be very limited to your university examinations alone. Of course, the content is going to help you in your university exams as well, but your understanding and knowledge should not stick to that one domain, and you got to evolve, you got to keep moving your limits, and you got to keep pushing your limits, you got to spread your wings, people. And this is exactly where we are coming into the picture. So guys, Pro MBBS is going to cover all the three first MBBS subjects, Anat, Physio, and Biochem. These subjects will be handled by expert faculties. By the next session and all, we'll be introducing our anatomy faculty and our biochem faculty, who have got breadth of entrance teaching experiences for years together. So guys, anyway, we'll be covering up all the subjects. We'll be finishing the portions usually 2 months before your university examination. So by and large, if we think that if you will be taking up your university examinations by November, we'll be wrapping up the portions by September itself, people. So these topics that we are going to discuss in Pro MBBS is going to be of a standard for a first MBBS student, and the standard is not going to be merely restricted to university exam. The priority and the target is going to be next exams, people. So I hope we could able to serve you better, and I'll be heading this program, guys. You have my email ID, you have my WhatsApp number, anything and everything, whatever is going in your mind, in case you find it anything difficult, you got any query or any problem to address, anything to be facilitated, I'm always there for you. So you have my word, guys. I'll make sure you learn Anat, Physio, Biochem in the best possible way, to the level of next, and not just alone to your university examinations, all right? I give you my word, it's my responsibility, people. So with this, let us take up today's topic, nerve action potentials and nerve conduction. At the end of this topic, we will be taking up few leaderboard questions, and you have the experience of the beautiful ADrPLEXUS leaderboard, and we'll assess ourselves how we have learned and what are the points that needs to be carried over in our mind. So guys, let's take up nerve action potentials and how does this action potential is getting conducted. So when it comes to nerve action potential, I want you to start with the fundamentals. You guys know very well when it comes to depolarization. When it comes to depolarization of a neuron, depolarization of a neuron is caused by sodium influx. And this influx of sodium happens by voltage-gated sodium channels. Even though it's a basic, even though it is a very primitive point, it's better we start our discussion from this point onward so that your understanding is going to be very holistic. Guys, remember, you require voltage-gated sodium channels, and these channels are very massive in numbers in a neuron. It is only through this huge numbers of voltage-gated sodium channels, sodium enters into a neuron, and that will cause depolarization. And you guys know very well, this depolarization is going to turn the neuron from resting state into functional state. Resting state into functional state. When it comes to functional state, I want you to be just very clear about the exact function of a neuron. We require neuron in two different responsibilities. These neurons are going to be either exciting a postsynaptic cell. Neurons are designed to excite a postsynaptic cell. It can be anything. It can be a skeletal muscle cell. It can be a neuron, whatever cell it may be. Neurons are either supposed to excite a postsynaptic cell or it is supposed to inhibit postsynaptic cells. So this is the ultimate purpose of a neuron. In order to execute this purpose, a neuron needs to get depolarized. For the process of depolarization, we require sodium ion to get into the neuron. And this sodium ion has to come in to the neuron in a huge quantity, for which we require voltage-gated sodium channel beaker. All right, so that's the fundamental point from which we are going to begin our discussion. Now, when it comes to a neuronal structure, let me take up a basic cell body and an axon, and this axon is going to end in axonal terminals or axonal endings. The cell body has got a nucleus over which we do see dendrites. All right, we could able to see dendrites attached to a cell body people. It is the dendrites that are going to get involved in a synapse with a presynaptic neuron. I repeat, we are going to see a presynaptic neuronal ending, and this presynaptic neuronal ending is going to end on a postsynaptic dendrite. So the one that you are seeing in green color is going to be presynaptic neuron, and the black one is going to be postsynaptic neuron. Action potentials from the presynaptic neuron is going to reach the terminals of presynaptic neuron, from where neurotransmitters are going to be secreted. The neurotransmitters that get secreted from the presynaptic neuronal terminal will touch upon postsynaptic dendrites, and hence we call this synapse as a dendro or axodendritic synapse. Axodendritic synapse, where axon of one neuron makes a synaptic connection with dendrite of another neuron. That is the importance of a dendrite. It's the region that receives neurotransmitter from a presynaptic neuron. When it comes to cell body has got a very important organelle inside the cytoplasm, and these organelles, doctors, are called as Nissl granules. These organelles are called as Nissl granules, and Nissl granules are the one that has got two components, and these two components are going to be mRNA and ribosomes. Ribosomes are seen attached with endoplasmic reticulum. So ultimately speaking, It is the Nissl granule which is going to be involved in synthesis of proteins. Every neurotransmitter, guys, is a protein, right? Every neurotransmitter is a protein. The synthesis of those proteinaceous neurotransmitters are taking place in the Nissl granules. When you come back, below the cell body, we are going to find axon. The beginning of axon from the cell body, this region, the beginning of axon from the cell body is called as initial segment of axon. This region is called as initial segment of axon. Initial segment of axon will usually be naked, which means there will not be any myelin cell over the axonal membrane. When you come a little bit lower, we are going to find myelin cells on the axonal cylinder. We are going to find myelin cells in the axonal cylinder, doctors, and these myelin cells are going to ensheath the axonal cylinder, thereby forming a gap in between two myelin cells, and this gap is nothing but node of Ranvier. I hope you know this. So that is the parts of neuron. We do have cell body to which dendrites are attached. We are going to see initial segment of axon, axonal proper, axon proper. Axon proper is myelinated by myelin cells. In between myelin cells, we are going to see node of Ranvier, and there is going to be synaptic terminals. Synaptic terminals, also called as axonal endings. And this axonal ending is going to make a postsynaptic, it's going to make a connection with the postsynaptic neuron beam. In this neuronal morphology, I want you to be very, very, very clear and aware of a very important point. Because it was a previous year entrance question that used to come in AIIMS, All India Institute of Medical Sciences exams, as well as JIPMER exams, Pondicherry JIPMER exams. By 2017, I hope you would have heard about an institute called as NIMHANS in Bangalore. NIMHANS stands for National Institute of Mental Health and Neurosciences, India's premier neurological institutes that has got only 3 post-graduation degrees. You can do straight away MCH neurosurgery after MBBS. You can do straight away DM neurology after your MBBS. You can take up MD psychiatry. So there are 3 specialty courses: neurosurgery, neurology, and psychiatry. A whole college dedicated only for neurological sciences. All right, people. They used to conduct an examination, and in that examination, there was a question which changed the concept of neuronal excitation once and forever. That was a phenomenal point and discovery that has been found out, and it was published in a journal. What is that? Before 2017, the idea was cell body, entire cell body is a single structure and entire axon is a single structure. What that discovery brought up and what the question in NIMHANS highlighted, there is a region in cell body that I'm highlighting right in front of you guys. You could be able to see this in your screen, all right? The region that I'm highlighting now with the green color, this region, right? And I'm highlighting this with an yellow color as well, so that you could be able to see this straight and clear. This region is called as axon hillock. This region is called as axon hillock. The important point here is axon hillock is a part of cell body and not axon. Please remember that. Axon hillock is a part of cell body. And you can clearly see in this region, in the axonal hillock region, you're not going to see any Nissl granules. That is another PG entrance question. So 2 points I want you to remember at the earliest discussion stage itself. Number 1, the terminal part of cell body where the cell body is going to fuse with axon is called as axon hillock. Number 2, at that region, axonal hillock, we are not going to find any Nissl granules. That is the way with which we identify axon hillock in a neuron if we take a section and we see it under a microscope. So guys, what's the importance of these segments and what's going to be the role of these segments in neuronal depolarization or generation of action potentials? You guys know when it comes to generation of action potentials, you need voltage-gated sodium channels. And let us see the distribution of voltage-gated sodium channels in a neuron. When it comes to the distribution of voltage-gated sodium channel, doctors, let me start from dendrites. The number of voltage-gated sodium channel in a dendrite is going to be very less, and the number is going to be simply 5 sodium channel per square micrometer. When it comes to cell body, which is also called as soma, this has got a voltage-gated sodium channel of simply on an average of 50 sodium channels per micrometer square. When it comes to initial segment of axon, a phenomenally important region, initial segment of axon, the number of voltage-gated sodium channel are going to be on an average of 1000 voltage-gated sodium channel per micrometer square. When it comes to node of Ranvier has got a record highest numbers of voltage-gated sodium channel on an average of 2500 voltage-gated sodium channel per micrometer square. So what is another important point you got to remember in this data? You have to by heart this value, doctors. This is a PG entrance question. When it comes to dendrites, guys, you already knew this. Dendrites is the region which is going to be involved in synaptic connection with presynaptic neuron. A presynaptic neuron is going to release neurotransmitters. It is this neurotransmitter that is going to combine with a postsynaptic neuron, depolarizing or stimulating postsynaptic neuron. And that region, the dendrite, which receives the neurotransmitter, has got very high number of ligand-gated sodium channel and not voltage-gated sodium channel. Please remember, dendrites have got extremely high density of ligand-gated sodium channels, doctors, and not voltage-gated sodium channels. Another entrance point, ligand-gated sodium channels are useful for production of local potentials and not action potentials, and not at all action potentials. So what exactly happens whenever a presynaptic neuron releases a neurotransmitter? This neurotransmitter is going to bind with, the ligand-gated sodium channel in the dendrites, opening up ligand-gated sodium channel in the dendrites. There is going to be very little amount of sodium entry into the dendrites. This very little amount of sodium entering the dendrite will gradually turn the neuron, the resting membrane potential will gradually keep moving positive. So with minimal entry of sodium through ligand-gated sodium channel in the dendrite, there is going to be very gradual increase in the potential of a neuron. Now, with that entry of sodium, the voltage-gated sodium channel in the cell body also will open, but that voltage-gated channel numbers in the cell body are very less. You know this, right? You have only 50 voltage-gated sodium channel, which is inadequate to generate an action potential. So the sodium that is entering through the dendrite, the sodium that is entering through the cell body, they both are decent enough to produce a local potential and not at all a depolarization. So where are we finding enough number of sodium channel to cause depolarization? It is going to be initial segment of axon. And my friends, remember this. This is the site of generation of action potentials. Initial segment of axon is going to be the site of generation of action potential. We can also call this as site of generation of depolarization. point number 1. So if you happen to see a question that asks you about the site of generation of action potential in a neuron, your answer is going to be initial segment of axon. Please remember, there are few channels, there are few neurons, and these neurons have got a very, very small initial segment itself. Imagine this, or look at me. Consider my head as a cell body, my neck as initial segment of axon, my body as axon proper. There are a few people or human beings you might be knowing who has got a very short neck. Right? You know, if someone is really obese, you can clearly see that they have a very short neck and their thorax begin quite, close to the oral cavity itself. So similarly, we are going to see some neurons which has got morphologically very small initial segment. In those neurons with very small initial segment, the number of voltage-gated sodium channel is again going to be very less. As a consequence, the problem is the number of voltage-gated sodium channel become inadequate, and hence initial segment is not going to produce action potentials. It is also not going to produce action potentials in some selective neurons which do not have decent length of initial segment. In those neurons, where else do we see production of action potentials? Exactly, it is going to be node of Ranvier. So what are all those kinds of neurons which has got a very short initial segment? As a consequence, there is going to be generation of action potentials in the node of Ranvier. It's going to be sensory neurons. Neurons that is taking part in conducting sensations. Please remember, neurons that is taking part in conducting sensations. They do have very small initial segment. They do have very small initial segment. As a consequence, there is going to be inadequate voltage-gated sodium channels. Inadequate voltage-gated sodium channel stoppers. As a consequence, in these sensory neurons, action potentials, also called as depolarization, are initiated at. This takes place at very first node of Ranvier, and that's your next entrance question. So remember, guys, if the question is specifically asking about a sensory neuron, I want you to be meticulously sharp. Your answer is going to be first node of Ranvier. If the question is vague and general about a simple neuron, that is all. Your question is not direct, your question is very vague. It is asking about a neuron in common. You got to go for initial segment as the site of generation of action potential stoppers. All right, good. So that's point number 1. Now, the essential criteria is the initial segment that generates an action potential has to convey or propagate this action potential, people. All right. The action potential that gets generated at the initial segment has to get propagated all the way to the axonal ending. Why is it so important? Because when it comes to axonal ending, guys, we will be covering this in a separate topic as neuromuscular junction. But as of now, have a basic idea. In the axonal ending, there are going to be neurotransmitter-filled secretory vesicles, and these secretory vesicles will be floating in the neuron. It will not come and touch the terminal. In order to get this vesicles close to the terminal for release, we require opening of voltage-gated calcium channels. Please remember, we need voltage-gated calcium channels to open so that calcium from extracellular fluid enters into the neuron, and this calcium is going to bring the vesicles very close to the axonal terminal. Once the vesicle reaches very close to the axonal terminal, there is going to be a damage in the cell membrane, people. The cell membrane is going to be slightly broken down. As a consequence, the neurotransmitter will be released into the synaptic cleft. So the release of neurotransmitter is dependent on calcium. Calcium comes via voltage-gated calcium channel. To open up the gates of voltage-gated calcium channel, we require action potential to be reached at the axonal ending. So the point you have to remember is, in order to release the neurotransmitters, we require action potential at the axonal ending. But where are we seeing genesis of action potentials? In the initial segment. So the action potential that gets generated at the initial segment has to be conducted all the way to the axonal ending for effective transmission of impulses to the postsynaptic neuron. How this is happening, we are going to study that. A very, very, very interesting and important point, doctors. Now let me take an axonal membrane. And this axonal membrane do have myelin cells. So let me take up myelin cells, people. 1, 2 myelin cells. And with this, the axon is going to end. And there is going to be synaptic terminal. We'll keep it that way. So for the sake of discussion, let me take up just 2 myelin cells and obviously 2 nodes of runway, doctors. What is the importance of myelin cell? Why do a neuron requires myelination? Remember this. The phospholipid bilayer of a neuronal cell membrane is not going to be very strong, or it is not going to be densely packed. And there are lots and lots and lots of a channel. And these channels are called as voltage-gated potassium channels, are very important channels you have to remember. Doctors, please remember. Voltage-gated potassium channel, this usually opens at depolarization. When voltage-gated potassium channel opens at depolarization, due to the concentration gradient, potassium tends to leak out of the neuronal membrane into the extracellular fluid. Potassium tends to leak out of neuronal membrane into the extracellular fluid. If you lose cations out of a neuron, what will happen? The neuronal intracytoplasmic voltage will become less and less and less positive. Even at times, the voltage will drop down to resting membrane potential. That will inhibit neuronal excitation, that will inhibit neuronal conduction of action potentials. So under any circumstances, we cannot afford to let potassium to slip out of the neurons. In order to prevent the leak of potassium, or in another way, nature has given us a beautiful cell called myelin that completely seals the opening, doctors. We are going to see myelin cell that completely seals the opening of voltage-gated potassium channel. As a consequence, the efflux of potassium is being prevented. All right, doctors, as a consequence, the efflux of potassium is going to be prevented. When potassium is not leaking out, all the potassium is going to stay inside the axonal cytoplasm. When more potassium stays inside the cytoplasm, there is going to be more positive voltage. And that is exactly how we are going to preserve the depolarization. And that is how the depolarization is going to get conducted. So guys, remember this. The importance of myelin, doctors. So what does myelin cell does? In one word, you have to strike this answer, and it's a very important question. It is going to be coming to you in your university exam MCQs, in your university exam short notes, saltatory conduction, in your university Viva voice examination. This is one of your, this is an examiner's favorite question, saltatory conduction. This is going to come to you when you take up your entrance examination as well. So in every front, this is an extremely important topic, my friends. So be clear. What does myelin cell do? Myelin cell prevents leak of potassium from neurons. As a consequence of this effect, myelin cells preserves depolarization, which is exactly why myelin cells are also called as, electrical insulator. You know this. Electrical insulator, doctors. Fine. So that's the importance of myelin in a neuron. All right. When it comes to genesis of action potentials, you guys know very well, generation of action potential takes place at initial segment of axon, where there is going to be good numbers of sodium influx. As a consequence of sodium influx, potassium will try to leak, and the leak of potassium will be prevented by myelin cells. In spite of the presence of myelin, there is going to be a very small amount of potassium leak. We can't prevent it. There is going to be a minimal amount of potassium leak, doctors. We can't prevent it. But the huge potassium leak will be prevented by myelin cells. That's for sure. Take it for granted. As a consequence, every single cation, instead of staying at one place, these cations are going to be progressively moving in the axoplasm towards the node of Ranvier. When all the cations reaches node of Ranvier, they create a positive potential in that node of Ranvier. As a consequence of genesis of positive potential in that node of Ranvier, there is going to be opening of voltage-gated sodium channel in the node of Ranvier. I repeat, you know there is going to be genesis of action potential at initial segment of axon, and this action potential at the initial segment is going to bring in cations, and those cations are prevented from getting effluxed out by myelin cell. Thereby they're all cations, right? They repel each other and they run forward in the axoplasm, and these cation goes to the node of Ranvier. We discussed that node of Ranvier is the region which has got highest number of voltage-gated sodium channel, right? 2500 sodium channel per micrometer square. All right, doctors? So, yeah. Now, just because these sodium ions are reaching the node of Ranvier, there is going to be intracellular positivity, and this intracellular positivity is going to open voltage-gated sodium channels in the node of Ranvier, through which we are going to find influx of sodium ions again. So this new influx of sodium through voltage-gated sodium channel in the node of Ranvier leads to genesis of new action potential in the first node of Ranvier. Again, potassium tries to go out. That will be prevented by myelin cell. Remaining cation repels among each other. As a consequence, these cations moves to the next node of Ranvier, and this will lead to positivity inside the neuron, opening of voltage-gated sodium channel, genesis of new action potential. So this is how every node of Ranvier is generating a newer action potential structures. Very, very, very important point. Remember this. Every node of Ranvier is generating new action potentials. All right. This kind of conduction where the action potentials are generated at every node of Ranvier is called as saltatory conduction, right? This is called as saltatory conduction. We got a chat box. We'll see. Video is lagging. All right. Just check it up, doctors. Yeah. Don't worry, guys. This is also going to be a, it's also going to be available in the recorded format as well. So once this, class is over, we'll be sharing this recorded video and the link to you. So don't worry. You are going to have a smooth understanding of this content. Don't worry at all. So let me repeat this point once again. Whenever there is going to be genesis of new action potentials at each node of Ranvier, we are going to call this conduction as saltatory conduction terms. All right, this conduction is going to be called as saltatory conduction. Now, I sincerely request you, my dear friends, please remember, saltatory conduction is not something that you have, as you have conventionally read, it is not jumping of action potentials. Please remember that saltatory conduction is genesis of action potentials, at initial segment of axon, and this further leads to genesis of action potentials at each node of Ranvier. genesis of action potential at each node of runway, doctors. All right. So that is what is called as saltatory conduction, doctors. Now, the point I want you to be meticulously clear is, when it comes to saltatory conduction, please remember this. It takes up three factors. Number one, it requires a diameter of axonal cylinder. When the axon is bigger, you can able to have more sodium inside, and that will increase the positivity, and there is going to be better propagation. All right. So please remember this. With increased size of axon, there is going to be more sodium accumulation. As a consequence of more sodium accumulation, there is going to be increased conduction. So diameter of axon is directly related to conduction velocity. That's a very important point you have to remember, which is why bigger neurons have got a brilliant conduction velocity and thinner neurons have got a very, very, very slow conduction velocity. That's point number one, diameter of axon. Point number two is going to be axonal membrane resistance. Axonal membrane should not let, all right, please remember, axonal membrane should not allow the potassium to get leaked away. It should not allow potassium to leak. All right, and who prevents this leak of potassium? This is done by myelinations. So myelination is another very important factor that increases conduction velocity. So number one, diameter of axon, that increases conduction velocity. Number two, myelination, that increases conduction velocity. Number three, an extremely important factor called it temperature. You need to understand this, my dear friends. When it comes to temperature, remember this. With increased temperature, the axoplasm becomes kind of liquidaceous in nature. I'll give you an example. You have a gel, all right? You would have seen a gel, a toothpaste gel, aloe vera gel. Usually, protoplasm or axoplasm is a jelly, right? With increased temperature, the jelly state of axoplasm becomes kind of liquidaceous. Now, imagine this. You have two containers. One container is filled with jelly. The other container is filled with water. All right. Now, what I'm going to do is I'm going to take a goli. All right. I'm going to take a small balls and I'm going to drop these balls on these two containers. Through which container the ball will reach to the bottom quicker, or the goli will reach the bottom quicker? It's the one that consists of water and not the jelly. So the medium that is present inside the axonal cylinder, if it is kind of a jelly state, it will slow down the conduction. If it is very liquidaceous or watery, it will facilitate the conduction. That is exactly what happens in temperature. All right, people, when it comes to, no problem, doctor. The class will be there for another 15 minutes. That's all. Don't worry. All right. So when it comes to the temperature, please remember, increase in temperature. Increase in temperature is going to increase the conduction velocity. So three important parameters: temperature, myelination, as well as diameter of axon. These three parameters that I just highlighted is going to increase conduction velocity of neurons. Conduction velocity of neurons. All right, people. Yeah, that's the way with which action potentials are getting generated, and that's the way with which action potential is getting conducted. So I hope you are now clear with saltatory conduction and how this saltatory conduction facilitates a neuronal transmission of impulses, and what are all the factors that influences saltatory conduction. Number one, diameter of axon. Number two, myelination. Number three, it is going to be temperature, doctors. All right. In non-myelinated neurons, that's a very good question, Dr. Ganesh. Very good. When it comes to non-myelinated neurons, let's finish that point as well. See, the difference between myelinated neuron and non-myelinated or unmyelinated neuron is, in a myelinated neuron, the myelin cells are very, very, very thickly attached to the axon. All right, consider my fingers, or my palm is myelin, my forearm is the axon. The myelin cells will be very strongly adherent to the axon, so there will not be any space between the axonal membrane and the myelin. When it comes to non-myelinated neuron, please remember this, very important, and it's a brilliant question. There will be myelin cell, but instead of wrapping it very tightly, the myelin cells are loosely attached. A loosely attached myelin cell is what is called as unmyelinated neuron. It doesn't mean complete absence of myelin. It doesn't mean a naked neuron. Please remember that, right? So you're going to have a loose myelin. You're going to have a very loose myelin, doctors. It says that the myelin cells are kind of loose, but it is not very tight. So there will be prevention of potassium efflux to some extent, but not to the greater extent as we are seeing in a myelinated neuron. So when it comes to a myelinated neuron, doctors, all right, so I hope, Dr. Ganesh, you got your answer clear. When it comes to a myelinated neuron, the myelination is very stiff and tight. As a consequence, there is going to be a complete prevention of leak of potassium. When it comes to non-myelinated neuron, it is not very stiff, it is not very tight. There is going to be a partial prevention of potassium leak, but not complete prevention. All right, doctors? Yeah, that's a very important point. You have to remember that. Now, 2 points before we move on to the clinical condition called multiple sclerosis. Let me just give you 2 points, people. Number one, when it comes to central nervous system, that includes brain, spinal cord, cranial nerve 1, olfactory nerve, cranial nerve 2, optic nerve, we are going to see the myelin formed by oligodendrocytes. So central nervous system structures, brain, spinal cord, cranial nerve 1, cranial nerve 2 have got myelination done by oligodendrocytes. Peripheral nervous system, spinal nerves arising from spinal cord, cranial nerves 3 from oculomotor nerve all the way to hypoglossal nerve. The myelination is done by Schwann cells. It is done by Schwann cells, doctors. Remember this. All right. So central nervous system myelin is done by oligodendrocytes. Peripheral nervous system myelin is done by Schwann cells. 2 very important points. Why I'm taking you to this point is there is an extremely important clinical condition called multiple sclerosis. In multiple sclerosis, there is going to be autoantibodies. There is going to be autoantibodies that are developed against oligodendrocytes, which means it is essentially a central nervous system demyelinating disorder. All right, autoantibodies are developed against oligodendrocytes. So this leads to damage of oligodendrocytes. Along with damage to oligodendrocytes, there is also going to be a damage to axonal cylinder. So oligodendrocytes are getting broken down, as well as axonal cylinder is also getting broken down. What will happen to conduction? Conduction will be impaired. All right, remember this. So guys, remember, in multiple sclerosis, there is going to be a significant drop in conduction velocity. Drop in conduction velocity, doctors. Fine. What else you need to know in multiple sclerosis? Please remember, you know very well in a normal neuron, if a temperature is increased, all right, if a neuron is normal, if the temperature is increased, there is going to be a brisker conduction and a faster conduction velocity. When it comes to a neuron whose myelin cells are getting damaged, because the voltage-gated potassium channels are exposed and not covered by myelin cells, whenever there is going to be an increase in temperature, there is going to be faster travel of sodium ions and potassium ions because the axoplasm becomes very watery. At the same time, these cations can able to get out of the potassium channels, and this leads to a significant drop in conduction. Significant drop in conduction velocity. So friends, unlike a normal neuron, where increased temperature increases conduction velocity, a damaged neuron in multiple sclerosis will be manifested with drop in conduction velocity as and when the temperature is being increased. Very, very, very important point, guys. All right, there is going to be a drop in conduction velocity. This phenomena is a very important phenomena and a classical presenting feature of multiple sclerosis, and this is called as Uthoff's symptom or Uthoff's phenomena. One of a very commonly asked question in entrance exams. Please remember, a patient with multiple sclerosis has got a classical history, a classical presenting feature. They come to you and they say, doctor, whenever I go in a very hot environment, whenever I exercise a lot, whenever I develop fever, I do find unable to use my hands or unable to sense sensations, or my eyes are not giving me enough vision. So there will be some neurological deficit because of lack of conduction, especially when the temperature is increased. And doctors, please remember, this condition is called as Uthoff's symptom, one of a classical presenting feature of multiple sclerosis. So what else happens and how does this multiple sclerosis improves, right? How does multiple sclerosis improves people? If the voltage gates are leaky, then will there two-way exchange of ion? No, Dr. Ritikesh, there is not going to be two-way exchange because the channel and the gates will allow only single directional flow of ions, not the other way around. And please remember, it is a potassium channel, and when the gates open, obviously the potassium is going to move from high concentration to low concentration. Where are we going to find more concentration of potassium? In the cytoplasm or in the extracellular fluid? Obviously in the cytoplasm. So when the gates open, there is going to be movement of potassium from high concentration to low concentration, that is from the cytoplasm to the extracellular fluid. This leads to drop in intracellular voltage and the depolarization is arrested, which is why when it comes to demyelination conditions, there is going to be leak of cations, and this cationic leak is going to cause a drop in conduction velocity. Now, with chronic state or with the disease progressing, there is going to be a positive sign and that tries to improve the conduction velocity, doctors, and what it is. Let me take a neuronal membrane, and in this neuronal membrane, I'll give you a myelin cell, and this myelin cell is getting broken, and inside which there is going to be a voltage-gated potassium channel. I think you could be able to understand the color coding. If I draw in red, it is going to be voltage-gated potassium channels. All right. So because of this opening of voltage-gated potassium channel and breakdown of myelin, there is going to be a leak. In order to prevent this from happening, or in order to manage this condition, a normal voltage-gated sodium channel, which is supposed to be present in the node of Ranvier, migrates. Voltage-gated sodium channel migrates to the site of damage, and when potassium is leaking via voltage-gated potassium channels, this voltage-gated sodium channel tries to bring sodium into the neuron, trying to compensate the drop in conduction velocity. This is a protective mechanism. But will this happen at every time? No. It is a very basic attempt by degenerating neurons to recover out of that injury, but it is not very good. As a consequence, the patient will have an initial improvement in symptoms, but over a period of time, when this neuron is completely broken, when the axon is completely broken, this will lead to a complete drop in conduction velocity. It will lead to conduction failure. It will lead to conduction failure, doctors. And that is one of a terrible pathophysiology when it comes to multiple sclerosis. Doctors, I hope now you have that's good. Doctors, I hope now you have a clear idea of how action potential is generated, how it is getting propagated, the factors that influences conduction velocity. And once the factors influencing conduction velocity are clear to you, you know how multiple sclerosis is getting manifested. Autoimmune degeneration of oligodendrocytes. And this degeneration also will damage the axon. So there is going to be axonal breakdown. This will make the potassium channel to become open. More potassium will leak, especially with increased temperature. And that condition is called as Uthoff symptom. When the condition becomes severe, there is going to be a drop in conduction velocity. There is going to be complete conduction failure. Guys, that completes our today's discussion. And soon you'll be given the leaderboard questions in your app. So please open up your app and try to get your leaderboard questions answered. With every question being done, I will explain each and every question, why this is right answer, why this is wrong answer, and you will able to assess yourself. And that is how you got to train yourself for your entrance exams, doctors. So my dear friends, please don't stop now itself. Whenever you read something, make sure you assess, make sure you evaluate by solving questions, MCQs. So I think, our guys will be sharing the leaderboard questions to you in your app. So get ready with your app, people. Let us finish the leaderboard questions as well.
Doctors, we kindly request you to join. I've shared the link in the chat. You can click that link, or you can follow the WhatsApp group where you are, and you can join it's going to be a 5 question quiz. It's going to be very short. And we'll be having the faculty discussing the questions, but anything which is discussed, it has to be assessed for a long-term memory. And this is one of the core ideas of ADrPLEXUS. After every lecture, there'll be an assessment which will be helping you. So we'll be starting it, and we request one and all to click the link which is shared in the chat. Click the link and please join. Once everyone joins the link, we'll be starting the leaderboard quiz, and you can see how you're performing in each and every question. Dr. Prem, can I share the slide?
Yes, sir. Please, sir.
Yeah. You need to stop sharing.
Yes.
Okay. We'll be starting the quiz. Another 5 seconds. Request one and all to join. We are starting the first question. And the fastest to answer will get the bonus points. And that'll be the final leaderboard, which will be released after the 5th question. So we are starting the first one.
Good luck, doctors. Best wishes. We'll discuss up the answers. All right. Guys, Actually, node of Ranvier is the correct answer. I congratulate you people. Very good. Now, others who have taken initial segment as the answer, I just want you to, understand what went wrong in this option. Please remember, look at the question. The site of genesis of depolarization in a neuron that carries temperature. What is the temperature? Temperature is a sensation, isn't it? So touch, vibration, proprioception, temperature, pressure, pain. These are all sensations, my friends. So the question specifically asks you a sensory neuron, but rather than calling it as a sensory neuron straight away, the question will give you, will present the data in a disguise, and the hidden subtle point is it's a sensory neuron. And obviously you guys know very well, it is the node of Ranvier, especially first node of Ranvier, that is going to carry the sensation. So congratulations, 18 of you. Very good, very good. So yeah, we'll move on to the next question. Oh, fantastic. Fantastic. Very good. The right answer is C, guys. Axonal membrane resistance decreases conduction velocity. All right. You guys know very well, axonal membrane resistance actually increases conduction velocity, right? So let us take up option A. Depolarization is through voltage-gated sodium channel. It is a right statement. Your question says all of the following are true, right? Except you got to pick the wrong answer. All right. So option A is a right statement, true statement. Option B, voltage-gated calcium channels are necessary for ACH release. You saw that again, that is a true statement. Option C, axonal membrane resistance decreases conduction velocity. Not at all. Myelination, axonal membrane resistance increases conduction velocity. So that is a false statement. Obviously, C is the best answer. None of the above, not at all. Anyway, guys, beautiful, beautiful. You did well. Good, very good. So we'll move on to the third question. Oh, lovely. Good, good, very good, people. Very good, very good. So let's discuss the question. It's a very interesting question. This is exactly how you will be getting your next exam questions, guys. Even though it is a physiology concept, that will be embedded and that will be capsuled within a medical clinical condition. So you have to have the capability to decipher the basic point from a clinical picture. Anyway, it's obvious that it is a case of beautiful. It's a case of Uthoff's phenomena. It's a case of multiple sclerosis. So what is option A says? Potassium channel has no role in conduction abnormality. Wrong. It is the potassium channel that is causing the conduction abnormality. All right. So option A is wrong. Option B, sodium channels are responsible for improvement in conduction velocity. Exactly, yes. Why? As I told you, when the myelin cells are damaged and degraded, there will be naked exposed potassium channels. In order to compensate that problem, sodium channel from the node of Ranvier migrates to the damaged region and tries to bring in sodium. So when potassium is lost at one end, sodium will be compensated to get into the neuron, and exactly sodium channels are responsible for improvement in conduction velocity. Beautiful. B is the right answer. Kudos, people. That's actually a tougher question and you have nailed it. Beautiful. Coming to third question. Yeah, so we'll move on to the fourth question, doctors. Very good, Dr. Amsadharani. You're right. Multiple sclerosis and without Svirna. Very good. Ah, lovely. Fantastic. You nailed it, guys. Very good. So remember, in multiple sclerosis, the damage is seen either to axon or oligodendrocytes. There is no damage to dendrite. There is no damage to axonal ending. Certainly, there is no damage to cell body. All right. So it's just a damage to axon. If you got oligodendrocytes as an option, I want you to pick oligodendrocytes if it is given in the option, people. Since it is not given, you got to go for the second best answer. And in this options, axon seems to be the best fit among the given options. So axon is the right answer, guys. We'll move on to the next question. Oh, lovely. Fantastic. Wonderful. So it's very obvious, right? Multiple sclerosis is a condition where autoantibodies are developed against oligodendrocytes, but certainly not against Schwann cells. Brachial plexus or nerves that are starting from spinal cord, they are spinal nerves, they are myelinated by Schwann cells and not oligodendrocytes. So definitely the following structures can be seen affected by multiple sclerosis, except is going to be brachial plexus, because Schwann cells will be untouched in multiple sclerosis, guys. So that completes our discussion. Doctors, you will be, having the link of recorded video, and yeah, we have our winner here, Dr. Bibin Jude. Fantastic. Congratulations. Beautiful, guys. And Dr. Deepika, Dr. Reshwat Sai, Dr. Kavi Rutrika. Yeah, very good, very good. Dr. Amsadharani, everyone, people, Dr. Amu, fantastic. You guys nailed it. And I'm extremely happy that you got it, you understood, and you have actually did a very wonderful job. So guys, yeah, keep in touch, and we'll be, bringing up our anatomy faculty, we'll be bringing up our biochem faculty to you. We'll release the schedule as quick as possible, so you'll have an idea of what's happening and what's going to happen. You have my mail ID, you have my number. Anything, if you find difficult, you need any content to be taught, you need any content to be revised, we are always there for you guys. So, we'll be in touch, and it's our responsibility to make sure you learn well, you progress well, and you crack next with the best of the knowledge and mark possible. Guys, you'll be getting the recorded video, in a link format. So in case if your friends have missed it, kindly ask them to, listen to the video and ask them to get enlightened with this content. So guys, even the session is going to be over within seconds. In case if you got any doubts, please do take my number. It is 7904697566. All right. I'll share my number here.